Oh, hello, everybody. Welcome back again. We are moving on to talk about the structure of the DNA molecule and its role as the molecule of inheritance. Having talked about inheritance patterns and talked about the role of chromosomes, now we need to talk about the molecule within the chromosomes that actually represents the genetic information. And of course, unless you've been living under a rock somewhere for a while, you are aware that this is the DNA molecule. So we are going to talk a little bit about the structure of the DNA molecule and how the current model of the DNA molecule was developed. There is quite a story involved in that. Unfortunately, we don't really have the time to get into the details, but it is quite an interesting story. It would be a polite way to put it. A lot of skullduggery, underhanded behavior. A lot of bias and just plain bad human behavior involved in the story. But we can't really dig deep into that in this class. But if you are interested in finding out the backstory behind that, there are any number of references that you can look up. You can Google the story. It's not difficult to find on the internet. And learn more about how Watson and Crick eventually arrived at their model of the DNA molecule. It's quite an interesting story. But having looked at the structure of the DNA molecule, then we're going to focus mostly on its function. First, looking at how it is replicated, and we'll get into some detail. Unfortunately, the book does not do a really good job of getting into the detail of how that takes place, but we do want you to know more about it than what the book actually details. So you're going to find a lot more information here in the PowerPoint and the narrations and the videos that accompany that. And of course, you can find all the videos associated with any of the presentations in the resources chapter, and they are listed in the order in which they occur in the presentation. So it's pretty easy to find them as you go through. And then we're going to look at the processes of transcription and translation, how we extract information from the DNA molecule in a way that it can be used by the cell to assemble polypeptides, which by and large are the end product of genes that are coded within the chromosomes. Not necessarily always proteins and not necessarily a single protein. This is something that we have discovered over the course of time, and we'll touch a little bit on that as we go through this chapter. And then we'll look at what happens when the nucleotide sequence of the gene gets altered. We know that as a mutation, and not all mutations are created equal, as you'll see. So there's a lot to cover in this presentation. I've broken it up into sections. There is an introductory section. Then there's a section on the structure of the DNA molecule, then a section on DNA replication, then a section on transcription, a section on translation, and a section on mutations. And I believe there are a couple of others I may have missed, but we'll find them as we go through. So there's a lot to cover today, but fortunately it's convenient to break it up into sections so you don't have to sit here and watch the whole thing all at once. You can do that if you wish, or you can watch a section at a time or several sections at a time. However it appeals to you to take a look at this chapter, but make sure that you watch all of the sections because there's a lot of detail in here, and there's, as you might guess, information that will allow you to answer exam questions in the upcoming exam that covers chapters 9 and 10, which is coming up very quickly, in fact, in about a week's time, a week and a half's time. So we're getting really close to the end of the semester at this point. And that's something to bear in mind. Okay, let's get started then. So the book opens, or the current edition of the book that you have opens with a discussion of the Zika virus, which at the time that the textbook was written, that edition of the textbook was written, was the virus of interest and the disease that everyone was concerned about. In a previous edition, it talked about the flu virus, which is what my notes relate to. And in some sense, the flu virus is even more representative of the current situation that we find ourselves in with the coronavirus, because the flu virus, or a flu virus, that broke out around 1918. 1917 to 1918 is when we believe the first cases started to appear, although they weren't immediately reported as such. But certainly from 1918 through to around 1920, a flu virus that came to be known as the Spanish flu, totally incorrectly named, because in fact, first victims of the virus almost certainly were here in the United States. It just happened to be the way in which the world found out about this, what would become flu pandemic, was via the Spanish press, because the Spanish at the time were not involved in World War I and felt free to publish everything. Other nations that were involved in World War I, including the US, felt that they could not publish this for national security reasons, because it might affect the outcome of the war as it was continuing. So that flu is, of all the pandemics that we are aware of, going all the way back to the 1300s with the first occurrence of plague or Black Death, starting in Italy and spreading out through Europe. All of those pandemics pale in comparison to the flu virus of 1918, 1919, 1920, because it is estimated that some 30, that's the conservative number, up to perhaps 100 million people died of this flu. We don't really know how many people died, because in many countries, particularly in the third world, Africa, Asia, recording who died was not a particular concern. And we just simply do not know how many people actually were killed by this virus. But we do know that it was a massive number. And of all the pandemics, including the current one, this was the most major pandemic, the most deadly pandemic on record in human history, in recorded human history. And it was a virus. And what is ironic about a virus is that in terms of its structure, it's very, very simple. A virus is simply a small piece of genetic material. We would be interested in those viruses that contain DNA, but there are also viruses that contain RNA. So there are two different classes of viruses that we recognize: RNA viruses and DNA viruses. And in each case, that genetic material is encased in protein. That's it. That's what a virus is. It's not even a living entity. We don't really know how to classify viruses in the big schemes of life. We don't have an effective way of fitting them into the classic way of classifying organisms, because they're simply not made of cells. They don't do anything that an organism does. About the only thing that they have in common with organisms is that they have genetic material, but it's not exclusively DNA, which by definition is how we describe cells. Cells have genetic material that is DNA. And yet viruses are the things that we are most concerned about, because it is very, very difficult, as you have no doubt become aware, as you've had to sit at home because of the COVID virus. It is very, very difficult to combat them, because you can't use antibiotics against them. And in fact, if you go to a physician with the flu and you ask for antibiotics, a physician should quite rightly say no, because antibiotics will not work against a virus. Antibiotics only work against living things, specifically bacteria. And so you can't treat a virus infection with antibiotics. Now, if you happen to get a secondary bacterial infection as a consequence of your viral infection, then yes, you do need antibiotics, but only if you have a bacterial infection. Antibiotics will not work against a virus. They are not living. How then do they become so deadly? Well, as you have no doubt learned from the COVID virus, they infect cells. What they do is insert their genetic material, either the DNA or the RNA, into cells. And because, as we'll discover in this chapter, the way in which genetic material is read is universal. And the code that is used in reading that coded information is essentially universal. Any genetic material that is inserted into a cell from a foreign entity, whether it be a virus or from another cell or even from another organism completely, will be recognized and will be read, and a polypeptide or a protein will be created in response to that coded information. And that's how a virus gets itself replicated. But in the process, some viruses can make you incredibly sick because of the cells that they invade. Now, that's not true of all viruses. Not all viruses are deadly. Some viruses are benign. You get infected with them and you may not even notice that you've been infected. And again, we know with the COVID virus, some people simply get infected, but they don't respond, they don't show any symptoms. That would be the symptomless infection. That is sometimes true with different types of viruses. They just leave no fingerprint behind to let you know that they've been there, unless you go searching in the genetic material, in which case you may find them. And we know that over the course of human evolution, and perhaps even before our own species emerged, but perhaps during the time of our ancestors, viruses certainly infected somewhere along the way, because we find the remains of those viruses in the form of genetic material in our own genome. One of the things that might surprise you, and this is something we'll get into in the last two chapters that we are going to cover this semester, is that only about 3%, perhaps 5% at the most, of your DNA actually is coding for polypeptides and other features. It actually represents the true genetic material of your cells, about 3 to 5% of it. The remainder has often been referred to as junk DNA. Now, we're getting away from referring to it as junk DNA because we think there are regulatory sequences in there that regulate how other genes are expressed. And we might get into that in a little bit of detail in one of the last two chapters, because we talk about regulation of genes in chapter 11. But the point here is viruses have inserted themselves into our DNA. They constitute some of this non-coding region because obviously they're not coding for human genes. The good news is that that viral DNA has been inactivated. It's sitting there in an inactive form. And over the course of human history, that DNA has never become activated. So we know that it has been permanently deactivated. The DNA is still there because it's incorporated into our chromosomes. And when the chromosomes get copied, which we'll be talking about in this presentation, that viral DNA gets copied right along with it because it's right in the chromosomes. In the case of an active virus, that may also be the case. Or it may be that the DNA or the RNA is copied separately from the chromosomes. It really depends on the type of virus and the information that is encapsulated in the viral genetic material. It really depends on the type of virus. It has a lot to do with that. But the point here is viruses are quite simple, and yet they are quite deadly. And it all comes about because they possess genetic material, and that genetic material codes for polypeptides that can be produced by the cells that they invade. And that in turn has effects on not only the invaded cells, but other cells as well, because the virus particles that are produced, because that's the whole point of what the virus is doing when it infects a cell, it wants to get replicated. And so once it gets replicated, those virus particles will then emerge from the cell, in some cases leaving the cell intact, but in many cases breaking the cell open, which is when the virus starts to have an effect on you. What do those released virus particles do? Go out and infect other cells. And so it becomes a case of the virus replicating itself in the initial cells that it invades, but then the particles that have been created go out and infect more cells. And that's when the viral infection becomes really problematic, particularly if it's a virus that damages the cells. So yeah, we could talk about the flu virus, we could talk about Zika virus, we could talk about COVID, the coronavirus. We could talk about any virus that infects humans, or we could talk about any virus that infects any organism, because it turns out that all organisms are subject to viral infection. There are plant viruses that are exclusive to plants. There are bacterial viruses, and they have, in fact, a role in the story that we're going to be looking at when we look at the structure of the DNA molecule. Some of the evidence that confirmed that DNA truly is the genetic material of cells was as a result of using a virus as a experimental tool. And there's a very nice video that talks about that, and I'll point that out to you when we get to it. There are viruses that attacked animals, and as we've learnt in terms of the COVID virus, and we could look at the flu, any number of different flu viruses. Viruses also have the ability to live in some organisms as harmless viruses, but then move from one organism to another type of organism and create all sorts of problems. That's the history of the flu virus. So we have bird flus, swine flus coming out of pigs. The COVID virus and other related viruses we think came from bats. The Ebola virus possibly came from bats. That's where we think it came from in Africa. The HIV viruses, we know it in humans, came from chimpanzees. Chimpanzees have a similar virus called SIV, or simian immune deficiency virus. And that in turn probably came from monkeys that the chimpanzees had eaten, because chimpanzees, we know, are carnivores and they kill and eat monkeys. And in that way, they might have become infected with monkey viruses that ultimately became the progenitor of the SIV virus, which when it jumped from chimpanzees to humans, when humans came in contact with chimpanzees and probably ate chimpanzees, which is something that happens in Africa, the virus started to infect humans, and eventually we came to recognize it as the HIV virus. So we know that viruses often come from a host organism. In the case of many of the viruses that affect humans, they come from a host animal, but they cross or jump what's referred to as the species barrier and jump from one species to another. And when that happens, very frequently, the virus in the new species becomes very unfriendly because it's now in a host that it did not evolve with. Is it possible for the virus to change its behavior over time? Yes, we've already seen that with HIV. HIV is no longer anywhere near as virulent as it was when it was first recognized as the cause of the AIDS epidemic. When we initially recognized AIDS and we recognized HIV as the cause of it, it could be as little as 3 months from diagnosis until you succumbed to AIDS, and very quickly thereafter died of an infection, because AIDS is basically loss of your immune system and you become very susceptible to other infections. Now, even if you don't use medications, you can live for many years with the HIV virus because it's changed its behavior. It doesn't serve the virus any good to kill its host very quickly. It needs to leave its host alive at least long enough to allow itself to be transmitted to another host, because that's the only way the virus can survive. I know that sounds very clinical, but that's the nature of it. And of course, with medication, we now have the possibility for people to live a normal life, even if they are HIV positive. And it's very possible that somewhere down the road, the COVID virus will also change its behavior. We're seeing it changing its behavior now. It's becoming more infectious. The Delta variant is way more infectious than the original Alpha variant. And it may have a different set of symptoms. There's starting to be some news release that it's causing different symptoms as well. But ultimately, it is in the COVID virus's best interest, if we look at it from that perspective, to change its behavior and not to be as deadly as it has appeared to be, because the only way that it can survive itself is by going to new hosts. And that's the reason why getting vaccinated, creating what is referred to as herd immunity, is really important. Because if enough people can no longer be infected by the virus will disappear because it has a limited supply of hosts that it can affect. And when it exhausts those and there are no more infectable people because everyone's been vaccinated or has developed immunity from surviving the disease, then the virus simply goes away because it can no longer survive. It needs individuals who are unvaccinated and have not had the disease yet, have no immunity to it, in order for it to survive, because that's the only way it can replicate. It needs a host in order to be able to replicate. So if you've not thought about getting vaccinated yet, or you're having second thoughts about being vaccinated, think about it very carefully, because the only way that we can stop this infection is to make sure that enough of us can no longer host the virus, and then it will go away. And that's the only way we can make it go away, by there simply being insufficient new hosts for the virus to replicate in, and then it will simply die off and go away. Otherwise, we're going to have to live with it. Now, that's the simple alternative. And if we allow it to continue to replicate unchecked, then the new versions like the Delta are going to emerge. And at some point, if we're really unlucky, we may get a virus that is easier to catch, more deadly than the original, and can get around any vaccination that we've got. And then we'll be in real trouble. That's the nature of viruses, unfortunately. Okay, let's move on and start talking about the nature of the DNA molecule. It's kind of a gloomy beginning, but that's the nature of it, I guess. So when we start thinking about DNA as a molecule in cells, surprisingly, the presence of DNA in cells was known by around the 1860s or 1870s. The presence of DNA in chromosomes was not figured out until the early 1900s, but the role of chromosomes as the potential location of Mendel's genetic particles, what we refer to as genes and alleles today, was pretty well accepted in the early 1900s, because once people recognized the significance of Mendel's work, it was very easy to connect that to chromosomes, because in the 1880s, the process of mitosis was figured out, and in the 1890s, the process of meiosis was figured out through microscope observations. And in both cases, it was recognized that the only thing that passes from one generation of cells to the next, and in the case of meiosis, likely pass from one generation of organisms to the next generation, were these chromosomes, these structures that were being identified in cells undergoing mitosis and meiosis. And when people identified just how important Mendel's explanations of inheritance patterns were, the only connection that you could make to Mendel's inheritance patterns and meiosis were the chromosomes, being the only thing that passed from one generation to the next. In the 1910s, some work by an American biologist named Thomas Hunt Morgan working with fruit flies, confirmed that genes really are located on chromosomes. It was no longer just a supposition, but it was reality that chromosomes were the hosts of the genes, that the genes were actually located within the chromosomes. Today, we can actually take that one step further, and we can identify very clearly the actual physical location of a gene on a chromosome, because we can create, excuse me, genetic probes using a synthetic piece of DNA that will bind to the known nucleotide sequence of a gene. We put a colored tag on that genetic probe, and we then take a look to see where it binds to a set of chromosomes, and we trigger the colored probe by using ultraviolet light, because they glow, they fluoresce in ultraviolet light, and that clearly then identifies where on a particular chromosome and which particular chromosome a gene is located. So we can go a lot further now and really nail down the fact that individual genes are located on specific chromosomes. It didn't occur to people early on, however, that DNA might be the molecule of inheritance, because it was recognized that there were two components to a chromosome, DNA and protein. And when it started to become very apparent that inheritance was a far more complicated process than anyone might have guessed, that Mendel's explanation was only the beginning of the story, not the end of the story, and that there were far more complicated inheritance patterns, like the sex-linked pattern that we talked about, and many other patterns that we did not discuss. People started to think, well, maybe a complicated process like inheritance does not require a simple protein, a simple molecule like DNA, because it was known that DNA was only made up of 4 constituents, what we refer to as the 4 nitrogenous bases or nucleotides. It might require something that was more complicated. And in fact, proteins seem to fit the bill, because we know that there are hundreds of thousands of individual proteins in just a single cell, let alone in a whole organism. And those proteins come in many and varied forms. And so by the 1930s and 1940s, biologists were starting to think in terms of perhaps protein as a molecule of inheritance. Now, it's not as if experiments weren't done in the 1920s and the 1930s and again in the 1940s that seemed to support the idea of DNA as the molecule of inheritance. But there was such strong support for protein, and particularly some really big name chemists, Nobel Prize winning chemists, were supportive of the idea of protein being the molecule of inheritance, that really the biological world focused on that idea, that protein was the molecule of inheritance, not DNA. DNA was considered to be just too simple to be the molecule of inheritance, because it only came in one form, that form composed of 4 nucleotides. But that would all change in the 1950s, and particularly when one particular experiment was published in 1952. So before we get to that experiment, let's just review what we do know about the structure of the DNA molecule and its companion molecule in the process of storing and utilizing genetic information, RNA. So you've probably heard of both of them, DNA and RNA, and the names tell you something about the molecules. And in fact, one of the things that you need to be able to do is to differentiate between the 2 molecules. And the name tells you part of the story. So DNA, for instance, stands for deoxyribonucleic ribose. So once you see that name with the OSE, you should recognize that that's a sugar. And so the constituent nucleotides that make up DNA contain deoxyribose as one of the components, because all nucleotides have a sugar as one of the three components, and the sugar represents the central part of the nucleotide with other things attached to it. So DNA is deoxyribose nucleic acid, reflecting the fact that the nucleotides do actually have an acidic property. So what does RNA stand for? RNA is ribose. Nucleic acid. So at the heart of the RNA nucleotides is the sugar ribose. So you can see that the sugars are very, very similar. In fact, the difference lies at what is known as the number 2 carbon in the ribose. Ribose and deoxyribose are both 5 carbon sugars. So it's at the number 2 carbon position that the difference lies. In deoxyribose, it's a hydrogen attached to that carbon. In RNA, it's a hydroxyl group, an OH group. That's the difference between the two sugars, but that changes the chemical properties of both molecules. So immediately now we have at least one difference between DNA and RNA. There are in fact 3 significant differences between DNA and RNA. That's number 1. Both of them contain 4 nucleotides. So if you're looking for something, a similarity, something that would be similar is that they each contain 4 nucleotides, but they don't contain the same 4 nucleotides. And we'll talk about what the 4 nucleotides in each of the 2 molecules are a little bit later. But you can see here the letters for DNA. They're already telling you the letters for DNA, A, C, T, and G. Now, those letters, strictly speaking, refer to one of the 3 components of the DNA nucleotides, or any of the nucleotides, what is referred to as a nitrogenous base. It is, in fact, what differentiates each of the nucleotides from each other. So A stands for adenine, C stands for cytosine, T stands for thymine, G stands for guanine. And these are the 4 nitrogenous bases that are attached or are found in the 4 different nucleotides that make up the DNA molecule. 3 of those letters are the same for RNA. So A, C, and G, so adenine, cytosine, and guanine, are also nitrogenous bases found in nucleotides in the RNA. The T, the thymine nitrogenous base, is not found in RNA. That is replaced by a very similar nitrogenous base. It's almost identical. Again, it's a very, very subtle difference called uracil, or U. So you're going to find that the letters for the RNA are going to be A, C, G, and U. And the U performs the same function in RNA as T performs in DNA, as you'll see. So in fact, I've just given you the second difference between them. Now, one of the things that we know is that these molecules are known as polynucleotides. But DNA and RNA are referred to as polynucleotides. So the note says this, but I'm going to put this in my own terms as well, just to reinforce this. So when you link the nucleotides together, it's the sugars and the phosphate and the sugar and the attached phosphate, and I'll show you a diagram that illustrates how this takes place, that link together to form what is typically referred to as the backbone of the molecule. And then the nitrogenous bases, the A, G, T, C, and U, point off to the side, I guess. RNA is a single-stranded polynucleotide. And DNA is double-stranded. So an RNA molecule always consists of a single strand of nucleotides or a single polynucleotide. And DNA consists of two strands of nucleotides or a double-stranded polynucleotide. So this animation, and again, you can find this in the resources page. So let me just write this in here, check the chapter. Resource page. The animation walks you through this, but basically we can look at this still shot from the beginning of the animation and we can see this. So you can see that there are two strands of nucleotides here. So here's one strand and you can trace that up. And here's the other strand that makes up the DNA molecule and you can trace that up. So this is the double-stranded nature of the DNA molecule. Now, there's something that you need to be very aware of as far as the double-stranded nature of the DNA molecule is concerned, and I'll point that out now, and we're going to mention this again when we talk in more detail about the structure, and when we talk in more detail about the function, both in replication and transcription and translation. These two strands are identified as being anti-parallel. So we can actually identify distinct ends to the molecule. And those ends come about because of the way we label the carbons in the sugar molecule. So there are 5 carbons in the sugar molecule, because that's what a ribose is, it's a 5-carbon sugar. And each of those carbons has a number, 1, 2, 3, 4, and 5. And the key numbers here are 3 and 5. But because there are also carbons in the nitrogenous bases, the organic chemists, when they figured out these structures, wanted to differentiate between the carbons in the nitrogenous bases and the carbons in the sugars. So they put a little tick mark, or what is called a prime mark, against the numbers for the sugars, the carbons in the sugars. So that's why you'll see this sort of thing. And we say that that is 5 prime. So if this is the 5 prime end of this strand, at the other end, and if we follow the strand through, we trace it. So we're going through this one. Up here at this end, it's going to be the 3 prime end. But because these two strands are anti-parallel, the other strand is not 5 prime at this end. It's 3 prime at this end. And so when we follow this one up, the other end is going to be the 5 prime end, the other strand. And that relates again to the way in which the sugars and the phosphates link to each other to form the backbone of the molecule. The nitrogenous bases are always attached to carbon number 1, in case you're curious about that. And they stick out into space. Now, in DNA, the nitrogenous bases in the two strands actually interact with each other. They form hydrogen bonds, but they only do that in very specific ways. So an A will always pair up with a T, and a G will always pair up with a C because of the way they form their hydrogen bonds. And in fact, it's this anti-parallel nature of the two strands that actually allows the nitrogenous bases to pair up. So we refer to this as the A to T and the G to C. We often refer to this as base pairing. Or the other term you'll find for this is complementation. We say that A is complementary to T and G is complementary to C, so that the two strands are said to be complementary strands, and you'll see that terminology used a lot. Now, in RNA, it's single-stranded, and typically you'll see it listed as 5' to 3', just the same. Those numbers become important when we start thinking about transcription and translation, because the product of transcription is an RNA molecule referred to as messenger RNA or mRNA, and that is in turn translated, meaning this information contained in that messenger RNA is translated into a sequence of amino acids that represent the polypeptide or the protein that's coded for the gene. And the idea of 3' and 5' becomes absolutely essential to understand in talking about DNA replication, in talking about transcription, and in talking about translation. So knowing where that comes from is also important. So this gives you some clues as to where that notation comes from, because that becomes important in your understanding of the function of the DNA molecule. So the key experiment that finally convinced everybody that DNA truly is the molecule of inheritance. It's the information containing molecule that all cells, all organisms use as their genetic material, was carried out by Alfred Hershey and Martha Chase. I had to stop and remember who was who. It's Alfred Hershey and Martha Chase. And this is often referred to as the Hershey-Chase experiment because of the two individuals that did it. Alfred Hershey was the head of the lab, and Martha Chase was a graduate student in his lab, and this formed part of her PhD work. And this is the experiment that used a virus that attacked bacteria, referred to as a bacteriophage. It's a virus that attacks bacteria. The specific virus was referred to as the T2 bacteriophage. It's a DNA virus. So it contained the two molecules that were being considered as the molecule of inheritance: a DNA molecule, the genetic material of the virus, and its coat of protein that packaged the DNA. So in that virus, there is DNA and there is protein. And Hershey and Chase figured out a way to separately label the DNA and the protein of these virus particles using radioactive isotopes that would uniquely label the DNA and uniquely label the protein, so that they could tell which part of the virus was getting into the bacteria when the virus infected E. coli. And they were able to show definitively that the only component of the virus that entered the bacteria was the DNA. And therefore it must be the DNA that was directing the bacterial cell to make virus particles, because it was able to understand that genetic material, the DNA molecule that was coming from the virus particle. And by extension, because bacteria themselves have their own DNA, then it must mean that the instructions for a bacterium must be contained in its own DNA. But then you can extend from there and say, well, all cells have DNA, whether they be bacterial cells or eukaryotic cells, because of course we know eukaryotic cells have chromosomes in their nucleus, and one component of the chromosomes is a DNA molecule. So if DNA functions as the genetic material of a virus and bacteria can read it and respond to it to make more virus particles, and read the instructions from the viral DNA, that means that the bacterial DNA must serve exactly the same function by extension. It can't be bacterial protein that's serving as genetic instructions. It must be bacterial DNA. But then you can extend it further, because we know all cells have DNA in the form of chromosomes, and it must be serving exactly the same purpose wherever we find DNA. It must be the genetic instructions. And so when Hershey and Chase published the results of this experiment, and I'm going to put a note here, the Hershey-Chase experiment. When Hershey and Chase published this in 1952, it prompted everyone to go, oh yeah, DNA must be the molecule of inheritance, because there's no other way of interpreting this data anymore. And everyone immediately started looking at what was known about the DNA molecule and trying to get information about the DNA molecule. Now, as a consequence of experiments that had been done in the 1940s, organic chemists knew the chemical composition of the DNA molecule. They knew about the nitrogenous bases, they knew about the sugars, they knew about the phosphate groups, they knew what the basic structure of all the nucleotides were in a DNA molecule. An American biochemist by the name of Erwin Chargaff had done chemical analyses of the percentages of each of the nucleotides turning up in DNA from different species, and had realized that every species had different percentage ratios of the 4 nucleotides, indicating that each species had its own unique form of DNA. And every individual from a particular species had DNA that contained the same percentages of DNA, of nitrogen or of nucleotides in their DNA, which should have been a big clue to everybody that DNA was a really good candidate as a molecule of inheritance, but people were still pretty much focused on protein at the time. The other thing that Chargaff noticed was that the percentage of A pretty much matched the percentage of T, and the percentage of G pretty much matched the percentage of C in any particular DNA molecule that you wanted to look at. These became known as Chargaff's rules, and ultimately, once Watson and Crick published their model of the DNA molecule, became known as the base pairing rules. Remember on the previous diagram, I said A pairs with T and G pairs with C? Well, it was Chargaff's chemical analysis that first indicated that that was the way the nucleotides must pair up in a DNA molecule. No one paid a whole lot of attention to that, and even Watson and Crick, for a while as they were building their model, didn't even pay attention to it. I think they completely forgot what Chargaff had done. Part of it was Chargaff's fault, because he was not a particularly easy person to work with, apparently. And he had this nasty habit of rubbing people up the wrong way. And so people, I guess, largely ignored what he was saying in his research as a response to that. But eventually Watson and Crick recognized what Chargaff was saying about the percentages of the A's and T's and G's and C's, and incorporated that in as they built their model of the DNA molecule. And that's one of the reasons why they were able to build their model as quickly as they were, because they published their paper in 1953, 1 year after, actually less than 1 year after Hershey and Chase published their paper, about the DNA molecule as the molecule of inheritance based on their bacteriophage work. So how were Watson and Crick able to jump on this really quickly? Well, it turns out that Watson and Crick were not biologists. They had no real interest in biology at all. One was a chemist and the other was a physicist. So really no interest in biology at all. But they both realized that whoever was able to crack the structure of the DNA molecule once Hershey and Chase published their paper would almost certainly get the Nobel Prize. And so they formed a partnership to work on the structure of the DNA molecule, and they started working on it for a little while, but were told fairly soon after by the head of the laboratory that they were working in to quit. Because that was not what they were hired to do. But once it became really obvious that the Hershey-Chase experiment had really nailed down that DNA was most definitely the genetic material, they were finally able to convince the head of the laboratory to give them permission to go ahead and work on the structure of the DNA molecule. So that allowed them to get started again. Then they also had access to information that other researchers didn't have access to. And how they got access to that information is part of the darker side of the story, because they got access to that information in a very underhanded way. And basically today, if someone was to do that, they'd probably get kicked out of the science field altogether because of what they did. But at the time, it was not fully understood exactly how they got access to that information, and so they were not held accountable for that. Because by the time that people realized exactly what they had done to get access to that information, they'd already published their paper and had garnered a whole lot of support for the work that they had done. And the person who was most hurt by what they had done had unfortunately passed away as a consequence of being exposed to a lot of X-rays. We'll talk about that person in a few moments. But before we move on, here is what is chemically known, or was chemically known, at the point that Watson and Crick started their work. So here is the basic makeup of a nucleotide. At the heart of it all is the sugar molecule. In the case of DNA, it's the deoxyribose. The nitrogenous base that identifies the particular nucleotide is attached to the carbon number 1. That's the 1' carbon. Here's the 2' carbon, which is where the difference lies. The 3' carbon is what is going to link to the phosphate group on the next nucleotide. And that phosphate group is itself attached to the 5' carbon, which is sitting here, of the sugar. So perhaps I'll label these. So this would be 5' carbon, and this would be the 3' carbon. And so on the strand, this would be the 3' end, and this would be the 5' end. So at the 5' end, there's always going to be a phosphate group. At the 3' end, there's going to be an OH group, or we refer to as a hydroxyl group in case you've forgotten. And so you can see now that you've got these alternating sugar, phosphate, sugar, phosphate, sugar, phosphate that builds the backbone. And here is the nitrogenous bases, the different nitrogenous bases sticking off the side, attached to carbon number 1, excuse me, of the appropriate sugar. So this is just reiterating the basic structure of the nucleotides, telling you about the name. I've given you all of that. The nature of the nitrogenous bases. So this is not something that you really need to delve into in more detail, but just note that the two of the nitrogenous bases have a single ring structure. 2 of the nitrogenous bases have a double ring structure, and that would create a problem for Watson and Crick for a short period of time, trying to figure out how the nitrogenous bases linked the two strands together. Because once they got access to the data that I was referring to, the stuff that they got in sort of an illegal way, they were quickly able to figure out that it was a double helix consisting of two strands, which meant that there had to be some form of connection between the two strands, and it became obvious that they had to be in the form of the nitrogenous bases. Their initial idea was that two A's linked to each other, two G's linked to each other, two T's and two C's linked to each other across between the two strands, but that very quickly became unworkable because it was quite clear that the molecule was of constant diameter, and seeing as how C and T are single ring, they would form shorter rungs than a pair of A's or a pair of G's, the double rings. The only way to make a rung of consistent length out of a single ring structure and a double ring structure was to think in terms of a single ring and a double ring linking together. But then the question became which? Was it a T with a G and an A with a C, a with a T and a G with a C? What were the pairings? And that's where Chargaff's observations, the percentage of A and the percentage of T in any DNA molecule were essentially the same, and the percentages of G and the percentages of C were essentially the same in any DNA molecule. That let Watson and Crick know that the pairings had to be A with T, which was a single with a double, and G with C, which is a single with a double. And so that's why those pairings exist, because of the known structures of the nitrogenous bases and Chargaff's observations about the percentages. So Watson and Crick were not working in a vacuum. They had access to all of this information as they started their work in 1952 and on into 1953. It would be, and again, this is all just laying out the differences that I've already talked about between DNA and RNA. So this is showing RNA as a single-stranded molecule. So what Watson and Crick really needed was some physical information about the DNA molecule, and that would come from Rosalind Franklin, who worked at King's College. Now, the reason why they were able to get access to this was because Watson was a good friend of Maurice Wilkins, who was arguably a colleague of Rosalind Franklin. The two of them had an ongoing disagreement about exactly what was the nature of their working relationship. And Wilkins, one day when Watson came down to visit from Cambridge, showed Watson a photograph. And I don't have the actual picture in here, which is a shame. The textbook doesn't actually include it, but I have the picture elsewhere, and maybe I can include that somewhere. But Franklin and one of the students that worked with her had created an X-ray image of the internal structure of the DNA molecule. And in fact, Franklin was so good at doing this sort of thing from her work with proteins that she was able to produce an X-ray image that was much clearer than anyone else around the world could produce, including someone in the United States, a researcher by the name of Linus Pauling, who wrote the book on chemical bonding, literally wrote the book on covalent bonding and ionic bonding, and received the Nobel Prize for it. And he had started working on proteins and doing X-ray imaging of proteins. And he was one of the people who was actively promoting proteins as a molecular inheritance and limited people's interest in DNA for a long period of time because, after all, he was Linus Pauling. He wrote the book on chemical bonding. He got a Nobel Prize for that. He was a protein chemist. He would actually go on and get a second Nobel Prize. He would get a Peace Prize for his work trying to limit nuclear weapons and ban nuclear weapons. So he's one of several individuals who have received 2 Nobel Prizes over the history of the Nobel Prizes. So he was a big deal, obviously. But he was relying on X-ray images that were nowhere near as clear as Franklin's. And Franklin's image showed a couple of things. One of the reasons why Pauling missed out, and potentially Watson and Crick could have missed out, was because the initial thought was that DNA was a triple-stranded molecule. But Franklin's X-ray image said, no, it's double-stranded. So once Watson and Crick got this information via Wilkins, they immediately said, okay, it's double-stranded. We need to build a model that's double-stranded. That makes sense. It also indicated quite clearly that there were cross-links between the two backbones. And so having given it some thought, they realized that the sugar phosphates built the backbones and the nitrogenous bases must form the links, the cross-links. And because it was a helix, because it had backbones, because it had cross-links, they could now start applying some of the chemical information they knew. And eventually they built a wire model. And this is the first model that they built. And this is on display at the Natural History Museum in London. You can go and see this. It's in a glass case. I've seen it. Of the original, this is their original model of the DNA molecule that conformed to all the chemical information and also to Franklin's X-ray image. And this is actually built to scale. So obviously it's highly magnified scale of the DNA molecule, but it is built to scale. So all of these sticks and balls and everything represent a scale model of the DNA molecule. And it was this that represented the basis for the paper that they would go ahead and publish in 1953 on the basis of all the information that they had gathered together. So the basic idea of a DNA molecule is that it's a little bit like a rope ladder. You've probably seen this analogy before. It's a little bit like a rope ladder that is then given a twist. The rungs of the ladder are the nitrogenous bases forming pairs that cross-link the ropes which are formed by the sugar phosphates, or the backbone that's formed by the sugar phosphates. You can illustrate the DNA molecule in a number of different ways. So this is the ribbon model. You can draw it this way as the atomic model showing the locations of all the atoms. Remember, wherever the lines meet in a diagram like this, there are carbons, and sometimes we don't even illustrate what's being attached to the carbon. And again, just to reinforce the numbering, this is going to be our 3' end because there's the OH group. So this must be the 5' end here. Over here, we've got the OH group. So this must be the 3' end. And down here with the phosphate group must be the 5' end. And then you can draw a space filling model, which requires a computer. You can't hand draw this, which is another way of representing it. So in 1953, they published this paper. In the same edition of the journal, the next paper was the next article was a paper written by Watson, Crick, and Wilkins that provided further information about the structure of the DNA molecule. And then came a paper written by Franklin alone, talking about her X-ray image and her interpretation of the X-ray image. So at this point, Watson, Crick, and Wilkins are reinforcing the fact that they are working together and reinforcing the fact that they are excluding Franklin. And this would become a really sore point for many years because Watson basically reduced Franklin to the role of a technician producing a piece of data that they were able to work with, which is so far from the truth that it doesn't even bear thinking about. Franklin was a very, very outstanding chemist and produced really good results no matter what she did. Once it became obvious what Wilkins had done, Franklin became so discouraged and so ticked off that she left King's College and went to work for the British Coal Board. But unfortunately, shortly thereafter, I think it was 1957, she was diagnosed with ovarian cancer, which almost certainly developed because she spent so much time in a room with a machine that was producing x-rays. And apparently she didn't practice any sort of caution around the x-rays. She was known to walk directly across the beam of x-rays, not to wear any protective gear, even though it was well known that x-rays were potential mutagens. And so as a consequence of developing ovarian cancer in 1958, unfortunately, she passed away. Come 1962, enough information had been developed to support the two ideas or hypotheses that Watson and Crick had floated in their 1953 paper about the role of the DNA molecule. In that paper, they had indicated that they felt that the information stored in the DNA molecule was based on the sequence of nucleotides. That would take about 12 years to figure out, but by 1962, it was becoming increasingly obvious that that was in fact the case. Enough information had been gathered to basically say, yes, it is the sequence of nucleotides. And in 1956, just 3 years after Watson and Crick published their paper, another paper was published. I'm having a senior moment. I'll remember who the authors were at some point. But in Messelson and Stahl, so it's the Messelson-Stahl experiment. You can look this one up if you want. It was published in 1956 that confirmed Watson and Crick's basically one-sentence speculation that DNA was replicated in a semi-conservative fashion, meaning that the two strands of the original molecule served as templates for forming the missing complementary strands, and as a result, you would get two new DNA molecules that contained one strand that was new and one strand that was conserved from the original DNA molecule, what became known as semi-conservative replication. And so in 1956, Messelson and Stahl published their paper confirming that in fact DNA replication was semi-conservative. So in 1962, it was basically considered that Watson and Crick's model had enough support to be considered to be accurate. And so the Nobel Prize Committee awarded the Nobel Prize for chemistry to Watson, Crick, and Wilkins. Now, as the note says here, Franklin, we think, we don't know for sure, we'll never know for sure, but we think Watson, Crick, and Franklin would have been the three people to get the Nobel Prize if Franklin had been alive at the time. The one hard and fast rule about the Nobel Prize is you must be alive at the time that it is announced that you're receiving the Nobel Prize. And in fact, a few years ago, the committee actually had to rule on whether that was the case or not, because they awarded the prize to a chemist who subsequently died after it was announced that he was receiving the prize, but before they had the ceremony in Sweden where the prizes were handed out, because there is about a 5-month separation between when the phone calls are made and the announcements are made of the awards and the ceremony where the awards are actually given out. And they decided that because he was alive at the time of the announcement, that yes, he was a Nobel Prize laureate. So that's why you at least have to be alive at the time of the announcement. Unfortunately, in Franklin's case, she died in 1958. The prize was awarded in 1962. For a long time, Franklin's role then was largely unrecognized, or at least not recognized for the significance that it truly held, in part because of Watson and Wilkins, not so much Crick. Crick and Franklin are actually quite friendly. Watson and Wilkins, not so much. But in more recent years, Watson has not covered himself in glory. He's still alive. Crick and Wilkins have subsequently passed away. Watson is still alive, but basically well out of the public eye because he has made, over the course of time, some very unflattering statements, not only about Franklin, but about the role of women in science in general. And it got to the point where people just basically said, that's totally unacceptable. And he actually was fired from his role as a university president. I think he was president of the university in Baltimore at the time, and he has subsequently not made any more public announcements. But Franklin's role in all of this has now become widely recognized. Unfortunately, just like Mendel, it does her no good because she's not alive to receive the congratulations on her contribution. But unfortunately too, that is sometimes the nature of science, that people don't get recognized for their contribution during their lifetime. So this animation looks at the basic structure of the DNA molecule. And it runs through the three models. So it shows you space filling, the tape version, and what is referred to as wireframe. We've seen those before when we were talking about protein structure many chapters ago. But this animation walks you through that. So you can take a look at this animation. Again, it's in the resources page. So you can see here that Watson and Crick did not really say very much about replication. As I said, it was a one-sentence note, and this is actually a word-for-word verbatim copy of part of that sentence. It immediately suggests a possible copying mechanism for the genetic material. That's it, that's all they said. And in terms of how DNA is copied. But as I said, people would go on and take that as a challenge to show that it was in fact what Watson and Crick were indicating, that it was semi-conservative. That was basically what they were implying here, even though they didn't state it in detail in this original paper. In the paper that they wrote with Wilkins, the second paper, they gave a more detailed explanation of the copying mechanism as semi-conservative. But in the initial paper announcing the 3D structure of the DNA molecule, this is all they said about the copying part. So that's a little bit about the structure of the DNA molecule and how it was arrived at. And we've taken a little bit of time going through that, probably a little bit more than I wanted. And I got into a little bit more detail about the backstory than I originally intended to, but I think it's good for you to know some of that, because it lets you know a little bit about the process of science. So it's time for a break. And as always, we have a concept check question that I will give you the answer to when we come back from the break. So here's the question. Compare and contrast the structures of DNA and RNA. Identify the three differences between the two molecules. I'll see you on the other side of the break with the answers.